Assalamu alaikum. Welcome to the great biochemistry lectures. In this lecture, we'll be talking about regulation of hemoglobin function. So the idea here is that last time we talked about the structure of hemoglobin, and we talked about how oxygen, when it binds to hemoglobin, it switches hemoglobin structure from the T to the R state. Now, there are other factors that can, or other molecules that can bind to hemoglobin, and they can also switch hemoglobin from one structure to the other, from a low affinity structure to the high affinity structure, or the opposite. And these factors are known as allosteric factors, since hemoglobin is an allosteric protein. So an allosteric protein is basically a protein that has different structures depending on what binds to it. These are known as allosteric effectors. Now, the idea is that a hemoglobin molecule, the hemoglobin is an allosteric protein, meaning that it goes through different structures, R and T. And these two different structures are regulated by binding to different proteins, or different molecules, These different molecules can bind to the molecule, and they can change its structure from, the active form or the high affinity form to the low affinity form. Now, these molecules do not need to bind to the same place where the substrate, quote-unquote substrate, or the original molecule like oxygen, for example, bind to. So they can bind to regulatory regions other than if we're talking about an enzyme, the active site. Okay. So in case of hemoglobin, they bind at places other than the oxygen binding site. Okay. Now, these are the effectors that we will discuss in this lecture. We'll talk about hydrogen ions, we'll talk about carbon dioxide, 2,3-bisphosphoglycerate, chloride ions, and carbon monoxide. And these are all known as heterotropic effectors. So oxygen is a homotropic effector because binding of an oxygen would affect binding of another oxygen molecule. So we're talking about the same molecule. So here we're talking about binding of these molecules would affect binding of a different molecule, which is oxygen in this case. So these are known as heterotropic effectors. So let's start talking about the hydrogen ions and what they do. So hydrogen ions are basically, we're talking about pH. And the idea here is that as pH goes down, affinity goes down as well. So the more hydrogen ions exist surrounding hemoglobin, the lower the affinity is. So you can see in here, this is at 7.4, that's physiological pH, so it looks like this, that's in green. So if you decrease the pH, what will happen is that the affinity decreases. And this is measured by P50, right? So the idea is that you would need more oxygen to get to 50% saturation of the hemoglobin molecule or hemoglobin molecules. Similarly, if you increase pH, that is, if you lower the concentration of protons, affinity increases. And this is known as the Bohr effect. That's after a scientist known as Bohr. So this is known as, so the effect of pH on oxygen affinity in hemoglobin is known as the Bohr effect. Same thing here. Okay, now, so it's important to understand the mechanism of the Bohr effect. How is it that protons stabilize the T state of the hemoglobin molecule? So it has to do with, primarily it has to do with histidine 146. This is the very last amino acid residue in the beta globin chain. So this histidine has the ability to form two important electrostatic interactions. The first one is with lysine on the opposite chain, that is in the alpha subunit. Okay, so this electrostatic interaction takes place between the carboxyl end of the amino acid residue, of the histidine residue, with the possibly charged R group of lysine. Now, at the same time, since the imidazole ring has a positive charge, it can also form electrostatic interaction with aspartate within the same polypeptide. Now, these two electrostatic interactions stabilize the T state of the hemoglobin molecule. Now, there is something quite interesting about the pKa value of histidine 146. So let me define first what pKa is. Okay, so the idea is that pKa is the pH when the concentration of the conjugate base is equal to the concentration of the acidic form of a weak acid. So in this state, the group is 50% protonated and 50% unprotonated. Okay. Now, what happens with histidine 146 is that the pKa is reduced from 8 in the T state to 7 in the R state, meaning that in the R state, it's easier for histidine 146 to lose a proton. So it loses it at a lower pH. So in the R state, histidine is not protonated, and as a result, it is not charged. So in the R state, histidine 146 cannot form this electrostatic interaction with aspartate. On the other hand, in the T state, the pKa is 8, meaning that it starts to lose this proton at a higher pH, meaning that at physiological pH, the histidine is mainly protonated and it forms this electrostatic interaction. Okay, so at pH 8, in the T state, histidine is 50-50. In the R state, at pH 7, it is 50-50. So basically, let's say at pH 7.5, the hemoglobin would be mainly in the T state because it is protonated, it is charged, it forms this electrostatic interaction, but in the R state it is not, because it loses this proton easily, so it is not protonated, and it's primarily in the T state, not in the R state. Okay, now I hope that this is clear. So basically, when the pH is low in peripheral tissues, the hemoglobin would mainly be in the protonated state. Mainly, it has this electrostatic interaction, and the protein is in the T state rather than in the R state. Now, the question is, where do protons come from? So in tissues, you have metabolism and you have the production of CO2. That's in Krebs cycle. So CO2 combines with water, carbonic anhydrase, forming the carbonic acid, which dissociates into the bicarbonate ion and protons. So the pH in tissues is lower than it is in the lungs. That's a result of metabolism, production of a lot of protons. So that would cause the protonation of histidine 146 and the formation of the T form. And that makes sense, because now in tissues, what you need is to have hemoglobin in the T state, in the low affinity state, so that oxygen is released in tissues. Okay? So we said that protons come from metabolism, the formation, generation of carbon dioxide in tissues, and that results in the formation of protons. So that's the first mechanism. Okay, so the idea here is that the more CO2 you have, the lower the pH. So that's pH of 7.6 at 25 torr of CO2. This is 7.4 when the pressure of carbon dioxide is 40 torr. When it is 60 torr, pH is now lower. It's 7.2. So that's the first mechanism by which CO2 can switch the hemoglobin from the R state to the T state in tissues. Now the second mechanism is formation of carbamates. So what happens is that CO2 itself can bind to the free alpha amino terminal groups of polypeptides, forming what is known as a carbamate. And this would create the formation of more negative charges. That is, you have more electrostatic interactions that can take place, and this stabilizes the T state of the hemoglobin molecule. So we have two mechanisms, right? The first mechanism is that CO2 can form hydrogen ions, lowering pH, and the second mechanism is that CO2 can bind directly to the hemoglobin molecule, stabilizing the T state. The question is, which one is more important than the other? Now, this was done in an experiment where pH is stabilized using buffers. Remember, we talked about buffers before. So pH can be stabilized, but CO2 is not. So in here, you have three samples. You have a sample where pH is 7.4, no CO2. And here you have pH of 7.2, no CO2. So right here between red and blue, what you're doing is that you're comparing the effect of pH, because that's the only variable. That's the only thing that is different. It's hydrogen ions, not CO2. And you can see, compare red versus blue, you can see that there's a shift in the saturation curve to the right, meaning that there is lower affinity when the pH is lower. Okay, that is, again, you need more oxygen to fill 50% of the hemoglobin molecule, to saturate 50% of hemoglobin molecules. Now, in the third sample, what we've done here is that pH is 7.2, but there is CO2. So you compare blue to purple. pH is the same. The only thing that is different is the concentration of CO2. And as you can see here, there's again shift to the right. So CO2 lowers affinity as well. And it turns out that 70% of the effect of CO2 is a result of forming protons. And 25% of the effect of CO2 is due to the formation of the carbamates, or what is known as the carbamino compounds. So CO2 is transported. How does it get transported? Well, it's transported as a bicarbonate ion predominantly. So 60% of CO2 is transported in blood in the form of the bicarbonate ion. So what happens is that CO2 leaves tissues, it goes into red blood cells. In red blood cells, some CO2 is bound to hemoglobin, but the majority is converted to the ions, to the bicarbonate ion and protons, and that's how, and bicarbonate ion goes out, and that's how CO2 is transported. So about 30% of CO2 is transported as a bound form to hemoglobin, and very small percentage of CO2 is transported in the form of dissolved gas. Now, remember, what happens here is that you have always formation of protons right here. And even though, and this is known as but even though there is formation of protons, there's no change in pH overall. And this is known as the isohydric shift, which is a result of hemoglobin acting as a buffer. Remember, we talked about the bicarbonate ion, and we said that it's the predominant buffering system in blood, but also hemoglobin plays a very important role whereby it can bind to protons. It can absorb protons. So hemoglobin plays a very important role in maintaining blood pH, a phenomenon known as isohydric shift. So let's talk about the other allosteric effectors. So there is a phenomenon known as the chloride shift. And this is an important phenomenon. So what happens here is that you have this transport of the bicarbonate ion in and out of red blood cells. That means that there is charge movement across the plasma membrane, and the difference in charge must be maintained. So what happens is that whenever you have exit of the bicarbonate ion out of red blood cells, this is balanced by entry of chloride ions. And whenever the bicarbonate ion gets into cells, the chloride ions would go out. So this is known as the chloride shift, and it maintains charge difference across the plasma membrane of red blood cells. So again, what happens here is that in tissues you have production of CO2. CO2 goes into red blood cells and it combines with water, bicarbonic anhydrase, it dissociates into the ions, bicarbonate ions and protons. Now the bicarbonate ion goes out, and the chloride ions would get in, again balancing the charge. Now here you have hemoglobin mainly in the T state. Part of it is that you have the Bohr effect, right? So you have the transition of hemoglobin from the R state to the T state, and that facilitates the dissociation of oxygen from hemoglobin, and oxygen gets into tissues. Now the opposite is true in lungs. So in alveoli, what happens is that the bicarbonate ion goes in, chloride ions would go out. Okay. Now again, bicarbonate ion forms CO2, and CO2 is exhaled. At the same time, you have hemoglobin being in the R form, so it can, with high affinity state, it can form, it can complex, or it can bind to oxygen atoms. So, question is there an effect for chloride on the transition of hemoglobin from the T to the R state and vice versa? And the answer is yes. So here's what happens. This is what chloride ions do. Once they increase in level inside cells, they form electrostatic interactions with the N-terminus of the alpha-2 chain, and chloride ions can also form electrostatic interactions with the R chain of arginine 141 of the alpha-1 chain. This arginine is the very last amino acid in the primary structure of the alpha-1 chain. Now remember that the alpha-1 chains are composed of 141 amino acid residues. So that increases the number of electrostatic interactions within the hemoglobin molecule, and that would stabilize the T structure of hemoglobin, lowering the affinity towards oxygen. So in tissues, oxygen is again released, and the opposite takes place in lungs. There is another important allosteric effector, and this is known as 2,3-bisphosphoglycerate. 2,3-bisphosphoglycerate is a byproduct of glycolysis. So it's not the main product of glycolysis, rather it is a byproduct. So in glycolysis, you have glucose being converted to pyruvate at the end, right? One intermediate is known as 1,3-bisphosphoglycerate. This 1,3-bisphosphoglycerate can be isomerized into a molecule known as 2,3-bisphosphoglycerate, which can then be converted to 3-phosphoglycerate. Okay, by removing the phosphate group. Now that's an important point in here. We'll talk about it in the next lecture in metabolism of erythrocytes. So what does 2,3-bisphosphoglycerate do exactly? Well, what it does is that it fits into the cavity of the hemoglobin molecule. So you have one 2,3-bisphosphoglycerate binding to one molecule of hemoglobin. Now note the negative charges that the 2,3-bisphosphoglycerate molecule carries. It carries multiple negative charges, and these negative charges would allow the molecule to form electrostatic interactions with lysine and histidine residues of the beta chain. So these interactions can stabilize the T form of the hemoglobin molecule. And 2,3-bisphosphoglycerate, what it does is that it shifts the curve to the right again. So it reduces the affinity. So note here, you have different concentrations of 2,3-bisphosphoglycerate. Here you have none at all. And note that the sigmoidal curve right here, but the curve is shifted to the left. So this has very high affinity. So it almost looks like myoglobin, which has really high affinity, but it's not there, but it's close. Well, if you have in normal blood, you have 5 millimoles of bisphosphoglycerate per liter. So you have the normal saturation curve. But if 2,3-bisphosphoglycerate increases to 8, note that the curve shifts to the right, meaning that you have low affinity hemoglobin in this case. And it turns out that individuals that live in high altitudes, they adapt by increasing the amount of 2,3-bisphosphoglycerates in their cells. Okay, so what happens is that if you go to a place that is at high altitude, the first 2 days you would have a hard time breathing as you're walking, but on the third day and further, your body adapts by increasing the level of 2,3-bisphosphoglycerate in cells. So this is a case right here where, again, that's normal curve for hemoglobin, and here if you have no 2,3-bisphosphoglycerate at all, note the difference in the P50 and the shift of the curve to the left. So I want you to appreciate the role of 2,3-bisphosphoglycerate in here. So what happens, let's look at the black curve. Now, at sea level, where the pressure of oxygen is 100 millimeter of mercury, about 100% of hemoglobin molecules would be saturated. Good. Now, when it goes to peripheral tissues where the pressure is 40, you have a decrease from 100 to about 55%. So we have a 45% of hemoglobin molecules releasing their oxygen. Okay, so that's 45% of oxygen. Now, in case there is no adaptation, no increased production of 2,3-bisphosphoglycerate. So at high altitude, right here, we have about 80% of hemoglobin molecules would be saturated. Okay, so once hemoglobin molecules reach tissues, again, the pressure is about 40, so we have 55% of hemoglobin molecules saturated. So that's about a 25% release of oxygen. So we're going down from 45% release to about 25% of release. Okay, so this is bad. That's not enough for peripheral tissues. So rather what happens is that you have increased production of 2,3-bisphosphoglycerate. You have a shift to the right. The curve shifts to the right. So now we're looking at the red curve right here. So the hemoglobin molecules would be saturated, and at high altitude, we're looking at point number 3, about 70% of hemoglobin molecules would be saturated. Okay, so that's little, right? But when it gets to tissues, notice the shift of the curve causes a lot of release of oxygen, where the pressure is about 40. So about 30%, there is 30% of hemoglobin molecules would be saturated. So we're talking about a 40% release of oxygen. Okay, so it's close to normal conditions right here. Now, this is again not a perfect solution, but what happens over time, there is more adaptation. The body produces more red blood cells, producing more hemoglobin molecules to compensate for this 5% loss of oxygen that is released. An important point about 2,3-bisphosphoglycerate is that its level goes down when blood is stored. So if a person donates blood and this blood is stored, even at 4 degrees, the level of 2,3-bisphosphoglycerate goes down within hours. So after 24 hours, there would be no 2,3-bisphosphoglycerate. Now, this shouldn't be a problem with normal individuals or people who are not severely ill, because they can restore the depleted levels of 2,3-bisphosphoglycerate within hours. But severely ill patients would be compromised because they would need 2,3-bisphosphoglycerate in their blood in order to successfully and easily release oxygen as hemoglobin gets into tissues. So for these patients, 2,3-bisphosphoglycerate would be added, as well as ATP would be added to these blood samples. Now, temperature also has an effect on affinity of hemoglobin towards oxygen. The higher the temperature, the lower the affinity. So what happens here is that, as you can see here, that's normal body temperature or almost normal body temperature. As it goes up, you can see that the curve shifts to the right. And that is a good idea. In a way, if you're exercising, for example, you want oxygen to be released easily in tissues. And if someone is sick as well, tissues must have high metabolic rate in order to fight the disease or whatever. So what happens is that increased temperature can also have an effect on the metabolic rate of red blood cells. That would increase the production of 2,3-bisphosphoglycerate, and that would facilitate oxygen unloading from or release from the hemoglobin molecule. So there is a dual effect or double effect of temperature on hemoglobin affinity. One is direct by affecting probably the structure or the interaction, the binding between oxygen and the iron of the heme molecule. And the other is that temperature would increase the metabolic rate of red blood cells. Now, there are other considerations as well. So let's first talk about fetal hemoglobin. Question is there a difference between fetal hemoglobin and adult hemoglobin? And the answer is yes. Fetal hemoglobin turns out that it has higher affinity for oxygen relative to adult hemoglobin. Okay, so that's a good idea because you want the fetus to get more oxygen. It's a competition, so the fetus wins the competition, right? And maybe that's why mothers, pregnant women, would gasp for air all the time, because fetus would take all the oxygen that they breathe in. So the thing is, why is it that fetal hemoglobin has higher affinity for oxygen? And the answer is that there are structural changes. And the main change is that there's a histidine residue in the beta subunit in the adult hemoglobin that does not exist in the fetal hemoglobin. Rather, you have a serine instead of this histidine. And the thing is, this histidine is important for interaction with the bisphosphoglycerate, the 2,3-bisphosphoglycerate. And serine cannot form these electrostatic interactions. So interaction between the 2,3-bisphosphoglycerate and fetal hemoglobin is weaker than its interaction with the adult hemoglobin, and that is why fetal hemoglobin has higher affinity for oxygen. And there is also an effect for carbon monoxide. Remember we said that carbon monoxide is toxic, right? It's fatal. And the reason why is that it has very high affinity for hemoglobin relative to oxygen. So that's oxygen saturation curve for hemoglobin, and this is the curve for carbon monoxide. You can see that carbon monoxide has much higher affinity. Well, it increases actually also the affinity of hemoglobin towards oxygen as well. So what happens is that whenever you have carbon monoxide bound to oxygen, the affinity is higher. Except that it's not a good idea because oxygen is not released. It keeps on binding to the hemoglobin molecule. So the saturation, so what happens is that not only that carbon monoxide occupies sites for oxygen binding, it also increases the affinity, preventing hemoglobin from unloading or releasing oxygen in tissues. And probably that's why it's toxic. So carbon monoxide can be fatal. Now the thing is, if the level of carbon monoxide in inspired air increases to about 1%, it can be fatal. Now, as a result of pollution due to factories, the concentration of carbon monoxide in blood can reach 1% in non-smokers. In smokers, it can get up to 10%. Now, if this concentration reaches to about 40%, as is caused by this 1% right here in air, it can be fatal to people. Now, these are the different chemical components that you can find in cigarettes, and they include things like things found in cleaners, candle wax, batteries, poisons, and so on. And I don't understand why people smoke. So, to sum up the lecture, we have the saturation curve for oxygen, okay? And it can shift to the right or to the left. If it goes to the left, affinity increases. If it goes to the right, affinity decreases. These are the different factors that affect affinity. Fetal hemoglobin, we talked about it. It increases, it has higher affinity as a result of this amino acid change from histidine to serine. Carbon monoxide also increases affinity, but it's fatal because it not only that it occupies oxygen binding sites, it also prevents the release of oxygen in tissues. These are the factors that shift saturation curve to the right, decreasing affinity, lower pH, the Bohr effect, the effect of CO2, we talked about two mechanisms, the effect of 2,3-bisphosphoglycerate, and we talked about temperature as well.